So yes, my name is Pralle Kriengwatana, and I'm very pleased to present this guidelines for you today. And again, just want to emphasize that it was a joint collaboration between researchers in KU Leuven and Odyssey and funded by the Vlaams Overheid and Department of Animal Welfare. So first, just an understanding of impacts on noise on humans. So noise is recognized as a major environmental health problem. It affects at least 20% of the European population. And statistics published by the European Environmental Agency in 2025 shows that not only are millions negatively affected by noise, but it also has real health impacts, such as being involved in premature deaths, cardiovascular diseases, and type 2 diabetes. So it's bad for humans. But animals, non-human animals, are also negatively affected by it. And we see this across taxa. So vertebrates and invertebrates, terrestrial and aquatic animals all respond in some way to noise. And many of these responses are, let's say, not beneficial for them. Now, in the world of conservation, they've mapped out kind of the pathway through which noise can affect animals. So they're both, sorry, I hope you see my mouse, my pointer. If
yes,
you don't,
we can see your
great.
pointer.
Great. So how it affects physiological responses and behavioral responses, and then its impacts on fitness. If you want to talk about lab animal welfare, I think we can focus on this. So what we see is that noise does affect physiological and behavioral responses that are important for lab animal welfare. For example, the effects on stress, it increases stress. It has effects on hearing, also in impairing immune function. In general, in terms of behaviors, sleep, reproductive behavior, foraging, territorial behavior, and also communication. And what that means is that noise thus potentially could have a negative impact on welfare, as well as the reproducibility of the science conducted on these animals. And this then undermines the goal of the 3Rs to reduce and refine research involving lab animals. Thus, the objective of the guidelines is first to provide evidence-based recommendations for managing sound for target species, here, rats, mice, pigs, chickens, and zebrafish. Second, it was to synthesize how the relationship between different types of sounds could be connected to stakeholder input regarding what they wanted and their priorities, and the feasibility of monitoring sounds and managing sounds, and to synthesize all of that into a single publication. Now, the guidelines are structured into different chapters. Generally, though, you can think of them as having two larger chapters. One is more of the background, so introduction to the guidelines, explanation of what sound is, basic sound perception in animals, and also sources of noise in laboratory facilities. The other half is research that we did to review the non-auditory effects of different types of sounds on animals, the results of the stakeholder interviews, and all of that put together into the recommendations and ideas for future directions. Okay, so now I will move on to this I will move on to explaining what is in the guidelines. So first, what is sound and how is it measured? So imagine someone is playing a guitar. Every time they strum or they pluck the string of the guitar, it vibrates. And this vibration leads to changes in air pressure that are picked up by our ears. This means that sound is a mechanical wave with changes in pressure caused by movement of particles back and forth along a fixed position. So it looks, if these black dots are particles, this is what it looks like. And then here is the resulting wave. This red dot here represents a single particle and its movement back and forth within a fixed position. When particles are densely packed, it's called compression. And when they're sparsely packed, it's called refraction. Now, sound is often studied, I forgot to mention here that for animals, like for a lot of animals, we only perceive the pressure variation. But for many fish and also insects, they're able to also pick up particle velocity. So they actually are able to hear the particle movement as well, which means that they are potentially hearing different things and things that we don't, since they have this extra way of perceiving sound. Now, sound is often studied by converting it into an image, which is a spectrogram. So here I'm showing you 3 spectrograms, all animal sounds. So A and B are sounds recorded in individuals under ideal lab conditions. So you can see that there is not much background noise in between the dark parts. C, on the other hand, is a recording in an outdoor condition, and each of the different colored boxes is a different species making noise. And you can see that it's much more complex in real life. And also the blue box indicates water sounds. So not only are there multiple sounds, but there are also multiple overlapping sounds. And from the spectrogram, you can get some important information about the properties of the sound. Some of the basic ones include time, which is represented by the x-axis, frequency, which is represented on the y-axis, and frequency refers to how fast the particles move back and forth. Faster movement, higher frequency, for us tends to correspond to higher pitch, so we perceive the sound as having a higher pitch. The darkness of the markings on the spectrogram reflect amplitude, or the amount of energy that is in that frequency at that time. As you can see from these spectrograms, most sounds are composed of many frequencies. So sounds with a single frequency, such as a pure tone, like a computer beep, are rare in nature. I want to talk a little bit about loudness, since this is something that has already been recommended in existing guidelines if they talk about sound at all. So loudness is a perceptual construct. It merges the physical property of the sound with what your brain perceives. An important point about this is that the brain will amplify certain frequencies and also de-amplify certain frequencies in a species-specific and potentially also experience-dependent way. So what I'm showing you now is two curves, weighted hearing curves. The Z curve, the Z curve, which is flat, it's not a curve, it's flat, and the A-weighted curve. And the A-weighted curve corresponds generally to human hearing. So the Z-weighted is basically a non-weighted curve. It just assumes that every sound is heard, every frequency, sorry, is heard with the same loudness. But in fact, for humans, you can see that sounds with quite low frequencies, they need to be much louder for us to hear compared to sounds within the frequencies between 1,000 and 10,000 hertz. So here, we are more sensitive to these sounds. They don't have to be, sounds with these frequencies don't have to be as loud for us to be able to hear them. And this is important because it means that two sounds with the same intensity, but different frequencies, may not be perceived as equally loud. Furthermore, we don't have standardized weighted curves for the different species, the different loud species that we work with. So it might be that what is loud to us is either very loud to them or not loud at all. And this is something we don't know. What we can know, however, is generally what their hearing range is. And from this, we can try to make some educated guesses. So for humans, our hearing range is between 2 and 20,000 hertz, so 2 kilohertz, with our most sensitive range here between 2 and 5 kilohertz. For the other animals, what's important to note here is that yes, the hearing ranges overlap, but for species like rats, mice, and pigs, they hear in the ultrasonic range, which means that there may be sounds in the environment that they are hearing and it's annoying them, but we cannot hear it. With chickens, it's the other direction. So they hear much lower frequencies than we do. So they hear infrasound. But the same problem occurs, is that they may be hearing things that we do not. So what we think is a quiet environment may actually be very loud and very annoying for these species. With fish, as I mentioned, yes, they seem to have the same hearing range, or at least are within our hearing range, but since they are able to detect particle motion, their sound environment might be very different than ours. So now that we have a general idea of what sounds they might perceive, what kind of sounds are in laboratory facilities that they might be hearing? Noise here is defined as unwanted sound. And there are several internal sources of noise, sources generated from the animals themselves, generated from humans doing the activities related mostly to animal care, equipment also related to animal care, but also general processes that have to happen in the building, such as ventilation and lighting and climate control. External sources can also exist depending on where the facility is situated. For example, if it's situated in a very busy area, you might have a lot of external noise of road, rail, traffic sirens, and constructions. If it's situated near other animal facilities, you may also get noise from that. Just to give you a general idea of how loud these different sounds are, for these aquatic species like zebrafish, it's estimated that what they're currently exposed to is about as loud as a motorboat. And these other sounds generated from these other sources can vary. Again, for reference, an average conversation is about 60 decibels. And so you see that building and technical systems, these are lower in how loud they are, but are also continuous. Whereas some of the animal, human, and equipment-related noises can be quite loud, but are more likely to be intermittent. So now I will talk about the review that we did and on the non-auditory effects of the different types of sounds. First, I want to introduce you to the outcomes of interest. So what effects were we looking for here? So we were looking at the effects of sound on reproduction, which we defined as litter size, hatching success, and or offspring survival. For immune function, we looked at glucocorticoids and their metabolites, but also differential leukocyte counts. So NL ratio or HL ratio, depending on if you are mouse or chicken, mammal or bird. Sleep, duration, and disturbances. But for social behavior, we restricted it to aggressive behavior, and we defined it according to the study, if they defined it in the ethograms as, for example, fighting, or if there are species-specific agonistic behaviors, for example, injurious feather pecking in chickens. For welfare, we focused on behavioral measures of anxiety or fear and abnormal behaviors, again, either defined in an ethogram or in standardized tests that are meant to evaluate fear, anxiety, and abnormal behaviors. For example, the elevated plus maze or open field test in rodents, and the novel tank diving test for zebrafish. So we used a systematic search protocol to search through Web of Science for the articles included in this recommendation. If you want to see the Prisma, we have that as well. But generally, we identified initially over 5,000 articles, and after lots of screening, 177 articles were included which met our inclusion criteria. The pie chart here just shows you the distribution across species, and this line chart shows you how many studies we found per species, per outcome. And as you can see, there are many in which either we found only a very few studies, 1, 2, or 3, or sometimes none at all. So I want to emphasize that there are gaps in our knowledge. So for many of these results, you have to take it with a little bit of caution since it's based on such few data. From these 177 articles, we categorize the sound types into these different categories. I will explain them, how we define them later on, but these are the ones that I will focus on in this talk. Then we had to quantify the evidence. So how do we know what effect sound is having on these animals? So for each outcome, we qualified the outcome as a positive, negative, no effect on the animal, or other. And other means, for example, that there are modifying variables affecting the outcome, such as sex or age. Please note that some studies compare the effects of multiple types of sounds and thus can contribute multiple data points. And then from these positive, negative qualifiers, we calculated a direction score. And the direction score ranges from +1, which is when all data points are positive, to -1, where all data points are negative. We also further calculated a confidence score, which you will see in the coming things, which is an asterisk, as having an okay confidence if at least 3 studies contributed data points to the direction score. On the next slide, what I will show you is a overview of how many studies we found per outcome, per positive, negative, no effect, or other effect. And this just shows you what you want to take home from this is, again, the gaps in our knowledge. So here, what we have is outcomes on the top, sound category on the left. It's too small to read, don't bother, but you will see it better in the actual printed guidelines. And on the right here is the different species. I understand this is not the type of mouse we're interested in, but it's just to differentiate it from the rat. So here, and the effects, the positive, no effect, negative effect, other, are on the bottom here. So what this shows you is that there are some in which we can there are some certain things where we can somewhat reliably say, okay, maybe this is having a type of effect, but there are others in which we have absolutely no information or very little information. So again, please exercise caution. Now I will move to the effects of broadband noise, in which we have the most data points for. So broadband noise are sounds described as broadband noises, which include things like generators, ventilations, fan, pink noise, white noise, and vacuum cleaners. With this figure, the gray represents no data. The white represents that there was no effect, so positive and negative are equal. The blue is a positive direction score, and a red is a negative direction score. So generally with broadband noise, what we see is that the effects of broadband noise tend to be quite negative across all the different outcomes and species, with a few exceptions. Also note that broadband noise these studies tend to play it very loud. So we cannot at this point separate the type of noise from how loud it was played. It's just, yeah, we just weren't able to do at this point. Note here that there can be some positive effects, and this seems to be driven by age, because the younger animals appear less affected than older animals. And for example, exposure during incubation can put of things to broadband noise has potentially positive effects on the animals later on in life. Okay. We then move on to music, which was our second largest category. So music, if the study described it, that they use music or radio, we included that as a data point. And in contrast to the broadband noise data, we see a very different picture, is that either there are no effects, animals don't care about it, or the effects tend to be quite positive, with perhaps some exception in sleep. Music tends to be played at a more decent level, so about 70 decibels, which is what you would probably play music at if you were listening at home. But also, I want to caution a little bit, even though it does look very good, that there may be publication bias. This is something we didn't look at in our recommendations. We move on to now urban noise. So urban noise are sounds described as vehicle noise, traffic, construction, general environmental noise. Very similar picture to broadband sounds. Again, with this weird blue, prenatal exposure in chickens seems to have more beneficial effects, and young chicks seem less to mind it less. Whereas again, in most other animals, the effects tend to be negative. Now we move on to narrowband noise. And narrowband noise are sounds described as composed of a single frequency, so for example, like the computer beep, or a very small range of frequencies. And what we see here is a lot more evidence in mice than in other species of a negative effect. So the mice studies suggest that acute and chronic exposure to loud narrowband noise increases anxiety and activation of the stress response system as measured by glucocorticoids. But it does it in a strain and circadian dependent manner. And it can also have negative transgenerational effects on offspring. You can read about it in the guidelines later. We now move to effects of conspecifics and heterospecifics. So these are sounds produced by animals either the same target, conspecifics, same species, conspecifics, or other species, heterospecifics. Now for these, I don't have any more of those nice plots because we have so few data points. So I will just describe a few studies here. So conspecific sounds, the effects are context and valence dependent. For example, playing sounds of distressed animals, distressed rats, to rats, increases glucocorticoids and anxiety. However, playing sounds of rats in a positive, happy rats doesn't seem to have an effect on rats. And same for pigs. Sounds of happy pigs don't seem to benefit pigs. With heterospecifics, we also see species-dependent responses. So if you play sounds of a predator, such as a cat, to rats, it increases glucocorticoids and anxiety, but doesn't affect mice. If we play sounds of humans to pigs, it has no effect on their sleep or aggression. So I think yeah, it's species specific and also relates to how they've interpreted the sounds. For example, humans may be quite benign. That's why pigs don't care. For animal rooms are sounds described as audio recordings of ambient housing environments. So they probably, for example, do contain broadband noises originating from fans, but also sounds from the animals themselves. In general, what we see is that lower levels of animal room noise, up to about 70 decibels, doesn't seem to cause prolonged elevation in glucocorticoids, but it could reduce resting behavior and alter aggressive behavior. Effects of alarm sounds. These are usually alarm bells, including things like a fire alarm. And in general, kind of acute exposure doesn't seem to have strong negative effects, but extended exposure does. So acute exposures are perceived as startling, but potentially not stressful. With ultrasound and infrasound, ultrasound are things that are too high for us to hear. Infrasound are sounds that are too low for us to hear. And ultrasound, loud ultrasound does affect rodents negatively, again, dependent on strain and sex. Infrasound also seems to affect rodents as well as zebrafish. Sorry, rats, not rodents in general, but rats and zebrafish. Just to make a quick summary of the findings now, the sound effects on welfare are highly context dependent. So it depends on the type of sound. It depends on the sound pressure level, which is loudness, whether it's predictable, the duration, is it chronic or acute, how old the animal is, and also the timing. With broadband noise, these studies tend to play it quite loud. It's frequently linked to elevated glucocorticoids, anxiety and fear behavior, sleep disruption, and also sometimes reduced reproduction. Urban noise, also play quite loud, similarly is associated with negative effects such as sleep disturbance and stress responses, especially in rodents. Dependent on frequency, timing, and age. Music, played at much more acceptable levels, can reduce stress and anxiety, but the effects will also depend on how loud it is, the duration, and the timing. For the other sounds, we have such little evidence that broad conclusions are not really appropriate here. But something we do want to take away is that there are no universal sound pressure level thresholds. So welfare effects cannot be predicted from how loud sounds are alone across species. The implication for that is that sound management should consider a lot of things other than how loud it is. It should consider species hearing, other properties of the sound, exposure patterns, and also the housing or experimental context. Okay, so now I'll move to input from stakeholders. So the goal of the input of stakeholders was to explore their priorities, ideas for solutions, and the feasibility of these solutions, ideas for monitoring, and also any questions that they had with regard to noise management. So one key finding is that there were large variabilities in the views and also some major knowledge gaps about managing noise. And not surprising with the knowledge gaps, if you remember that big figure with the circles that I showed you, we don't know. In terms of what sound features they thought were relevant to monitor, these included typically loudness, of course, the frequency, including ultrasound, whether the sound was continuous or discontinuous, how predictable it was, and possibly also vibration, especially in the case of fish. With noise monitoring, the stakeholders generally preferred continuous monitoring, but they didn't quite yet agree on the location of sensor placement, which weighting scales should be used, and whether to track short-term elevations in sound, so short-term peaks. In terms of mitigations, they felt that maintenance and staff are things that we can definitely tackle, whereas structural solutions are probably less likely due to cost and practicality. Some implementation concerns was that it should be these kind of sweeping guidelines are not so useful, but perhaps they should allow flexibility for each specific facility. and consider unintended effects such as the burden on staff and potentially, for example, music increasing stress rather than decreasing it. Okay, I will move on now to the recommendations. The first one being noise limits, because as I mentioned, noise limits are something that already exists in other guidelines. So here, 85 decibel A-weighted is already implemented as a limit in many facilities, and this is due to it being an human occupational health issue. However, this level may not be suitable for continuous exposure because it is calculated based on a workday, on a human workday. So some experts have suggested that 70 decibels over 24 hours may be a safer longer-term target. Consider, however, that even 70 decibels in rodents has been shown to affect the brain, sound perception, and learning and memory, although welfare effects of 70 decibels and these effects in them have not been qualified from a welfare perspective. So our recommendation is to maintain average facility noise below 70 decibels Z over 24 hours where possible. In terms of facility location and design, some of these I think are already generally implemented. So for example, to separate the sources of noise from the animal rooms. To use silent alarms when possible, and to make sure to check for devices that emit ultrasonic sounds. A field of research in which I think we could potentially learn from, which involves both sound minimization and hygiene, are in, I suppose, hospitals. And there are studies that show that there are these cleanable acoustic panels and ceiling materials that can be fitted without destroying everything to reduce reverberation and transmission between rooms and hallways. Of course, this is a larger cost, but just to let you know that some of these solutions do exist. To replace equipment emitting audible or ultrasonic noise during renovations, and specifically for aquatic systems, increasing the distance between the tanks and pumps can reduce noise, and to also consider vibration as well as sound. When it comes to training, education, and communication, we thought it's important that the facility define training, monitoring responsibilities, and management schedules, and to provide staff with training on procedures on how to reduce noise, as well as increasing their awareness of that species, such as fish, for example, are able to hear, and that noise does have negative effects on them. Communication here is also important. So for example, if you know that there's an unavoidable disturbance, communication of that knowledge to staff will also be important to help staff prepare. For example, changing the schedule so that it's less disruptive for the animals. In terms of monitoring sounds, ideally, facilities should identify potential sources of sound and define the monitoring strategies. As long-term unpredictable sounds appear the most detrimental, these should be prioritized. Technologies for continuous passive acoustic monitoring are available and cost-effective. However, the main challenge is data analysis and interpretation, especially if you want to monitor for long periods of time, potentially continuously. So periodic monitoring during equipment maintenance may be an option if continuous monitoring is not possible. And sensor placement is important. And for aquatic species, it should be in the water. You have, instead of microphones, hydrophones that are capable of measuring sound within water. When it comes to acoustic enrichment, music may mask ambient noise and provide sensory stimulation for lab animals. Based on our review, levels between 60 and 70 decibels do not appear to negatively affect rats, mice, pigs, and chickens. We can't say with zebrafish, though, because we don't have enough evidence. So if you want, our current recommendation is that if you want to play music, play it during active hours to avoid sleep. Studies on the music and sleep disturbance remain inconclusive. I'm missing a point here, which is that it doesn't seem to really matter what type of music you play. So across all the different, that chart where everything was pretty blue, there were lots of different types of music played. But it wasn't that some genres, for example, are particularly bad for animals. And it's also not the case that you can only play them classical music. So now to the last part, which is future directions. So one of the things I want to point out first is the limitations of the review that we conducted. First is that the outcome indicators are really narrowly defined. So for example, reproduction, there are so many other things we could have measured in reproduction, or for example, immune function, we took 2 indicators. So again, please keep that in mind. Also with the sound categories and characteristics, within the scope of this recommendation, we define the sound categories quite semantically. But sometimes the semantic label doesn't capture the properties of the sound itself. So what I think we should be looking in the future are for the characteristics of the sounds rather than a semantic category of the sound. So for example, things like the acuteness or infrequency of the sound, the chronicness or the frequency of the sound, frequent meaning how often it occurs, predictability, onset, how loud it is over what's currently in the background, whether it overlaps with biologically relevant, and also if it overlaps with hearing abilities. Because depending on these different characteristics of the sound and the animal, it could have a high impact or low impact on the animal's responses and behaviors, and thus potentially welfare. We also saw from a recommendation that there are many moderating variables. So the effects of strain, sex, and age are important, but understanding how they contribute are lacking. But we think it's important to look at these because it might help us to identify populations or phenotypes that are more resistant or vulnerable to noise. Finally, I think it's important to try to move noise management beyond noise limits. So beyond saying it cannot be louder than X. And I believe one way to do this is to use a soundscape approach to link sounds in the environment to animal responses. So this is borrowing from the field of soundscape ecology. So what you see here is a spectrogram where they've labeled, for example, those different sources of sounds and what's causing them. So from soundscape ecology, what you can do is to identify spatial temporal patterns. For example, if this sound occurs, is there a pattern in what other sounds also occur, and thus how the animal responds to these sounds? You can also, for example, quantify the relative contributions of different types of sounds. For example, they can split it into sounds that are originating from biophony, so animal-related sounds, geophony, things like water, wind, non-living sources, and also anthrophony, which is sounds created by humans. Yes, we're also animals, but we also create lots of sounds. So sometimes understanding the relative ratio or how much of these sounds are in the environment can give us some information on the soundscape itself. Acoustic indexes are also, I think, a useful way of approaching soundscapes and understanding how sound environments within the lab as a whole affect the animal rather than looking for very particular sounds, because these sounds will in any case exist in the context of other sounds in the environment. So it doesn't make sense to isolate them and look at them separately. And finally, once we find a way of characterizing these, we need to test the relationships between patterns and indexes to welfare indicators to see which ones we should be monitoring in the future. And if this may vary depending on the facility itself. So that is it. Thank you very much for your attention. I'm happy to receive any questions. If you have other questions that you would like to contact me in the future for, my email is there. As Axela mentioned, I'm also the director of the Acoustic Enrichment Interest Group. Our QR code for our LinkedIn group is there, but the aim of the group is trying to find enrichment strategies and how to understand how animals deal with sound and the effects of sound on health and welfare. So if you're interested, you're welcome to join the group and stay updated on the activities that we're doing in the enrichment acoustic enrichment interest group. Thank you.
Okay, thank you very much